In the following slides, I'm going to show my understanding of the paper Succession of Litter-Decomposing Microbial Organisms in Deciduous Birch and Oak Forests, Northern Japan. The study was conducted by Michiru Otaki and Shiro Tsuyuzaki, Graduate School of Environmental Science, Hokkaido University. This paper investigates the succession of bacteria and fungi in litter decomposition process in a pioneer and a climax forest. The key findings in this study are decomposition happened faster in the first year than the later years. Fungi was dominant in first year. In contrast, bacteria gradually became dominant throughout the period. Gram-positive bacteria won the succession over gram-negative bacteria. The forest types did not influence the trend of decomposition. Now, the materials and methods that were used in this study. The study was conducted in a pioneer forest that's dominated by birch and a climax forest that is dominated by oak. And the study site is situated in a lowland near the foot of Mount Toishi in Sapporo, Hokkaido, Japan. It was conducted for about 3 years, from December 2009 to August 2012. 3 different types of litters, birch, oak, and mixture of birch and oak, were put in litter bags on the ground surface of each forest. Litter bags were gathered several times each year, and various measurements were made in the laboratory afterwards. A generalized linear mixed effects model, chi-square test, still-Dewey test, canonical correspondence analysis, and Monte Carlo permutation test were used for generating different results. Now, the rate of decomposition in the forests. We see that the first year, about 66 to 69% of the initial litter mass remained in the both forests. In the birch forest, the decomposition was delayed for the mixed litter than the monocultural litters. We see that the rate of decomposition decreased over the time. To be more specific, by the end of the first year, the decomposition rate was 0.038 to 0.013 per day, but by the end of the study period, it was decreased to 0.004 per day. The decomposition rate was not different among the litter types and between the forest types, in a p-value greater than 0.05. In the slide, the ratio of carbon and nitrogen is shown. Carbon content, we see, did not change significantly between the forests and litter types, but it was found highest in the birch litter placed in the oak forest. Nitrogen concentration increased throughout the time, but was significantly different between the forests. Lastly, the ratio of carbon and nitrogen. Initially, the ratio was 52.8 for the birch litter and 57.3 for the oak litter, but at the end of the study period, the ratio decreased to less than 30. Here, the succession of fungi and bacteria is shown. The succession of fungi and bacteria is measured by the phospholipid fatty acids in the litter mass. So here, we find the fungal PLFA concentrations reached their peak in the first year, and it's the same for all the litter types and all the forest types. And bacterial PLFA raised steadily over the study period, irrespective of the forest types and the litter types. So, from these graphs, it is clear that in the early successional stages, fungi dominated the succession, but by the end of the period, bacteria gradually owned the succession. That means successional replacement occurred from fungi to bacteria. Here, the succession of gram-negative bacteria and gram-positive bacteria. The lowest concentration of PLFA is found for actinomycetes in both the forests and for all the litter types. Gram-negative bacteria reached their peak around 500 days of the study, but they followed a lower fluctuation afterwards. The PLFS in gram-positive bacteria, in contrast, gradually raised over the survey time, leading them to succession. The litter types influenced bacterial succession since the curves of PLFS are different among them in both the forests and in the litter types. So, to conclude, litter types and forest types does not cause a difference in the succession of microorganisms. This paper proposes that early colonizers, like fungi, promote the immigration and colonization of late colonizers, like bacteria. Or that is to say, the early colonizers facilitated the succession of late colonizers. This microbial succession was a key to understand the long-term litter decomposition mechanisms with increasing nitrogen concentration in the litter. This paper also suggests a possibility of a correlation between nitrogen concentration and bacterial succession. Thank you.